Hello, everyone. My name's Kaylee Lavelle, and today my presentation is on blood. Our bodies need blood to help deliver our nutrients and remove waste that lies within our cells. The heart is what pumps our blood throughout our body using blood vessels, and there are three components that make up our cardiovascular system, being blood, the heart, and its vessels. Blood is a connective tissue made up of cellular elements or formed elements, as well as an extracellular matrix or plasma. The cellular elements include red blood cells, white blood cells, and cell fragments referred to as platelets. The extracellular matrix or plasma is the liquid component of blood that allows the blood and cellular elements to circulate through the body within the cardiovascular system. Formed elements only live briefly, and therefore the body must form new blood cells and platelets quickly and continuously. 1 unit of blood lost will allow for plasma to regenerate within 24 hours, but it takes 4 to 6 weeks for the blood cells to be replaced, which is why you can only donate blood every so often. The replacement of process of plasma and blood cells is called hematopoiesis. Sorry. Functions of blood. Blood has many functions. Blood's primary function is to deliver oxygen as well as nutrients through the body while removing waste from cell bodies. Blood also has other functions such as defense, distribution of heat, and maintenance of homeostasis. First, we have transportation. We get nutrients from the food we eat, which are absorbed in the digestive tract. Most nutrients travel through the bloodstream to the liver to be processed before being delivered to body cells. The air we breathe diffuses oxygen into the blood, which then travels to the lungs before reaching the heart to be pumped to the rest of the body. Then endocrine glands release their hormones into the blood, which carries them to distinct target cells. And the waste that the body excretes is picked up by the blood to be transported to organs to be removed. One example of this is the kidneys. This would cause the kidneys to excrete the waste into bile or urine. So for defense holds white blood cells, and white blood cells protect the body from external threats like bacteria. Other white blood cells destroy internal threats like mutated cells that can multiply and eventually become cancerous. If vessels are damaged, blood platelets and proteins within the plasma will block the damaged areas of the vessel, protecting the body from blood loss. In this picture above is just our white blood cells. So lastly, we have maintenance of homeostasis. So on a hot day, as blood passes through the vessels in the skin, heat will be dissipated from the environment, returning your blood back to your body core at a cooler temperature. Whereas on a cold day, the blood is diverted from the skin to maintain a warmer body core. And that is why whenever you're hot, you sweat, and when you're cold, you shiver, like in the picture shown below. For the composition of blood, a test can be done to measure the percentage of red blood cells or erythrocytes in a blood sample called a hematocrit. Erythrocytes are cells made in bone marrow that contain a protein called hemoglobin, which transports oxygen from the lungs throughout the rest of the body. The blood sample is spun in a centrifuge to separate heavier elements from the liquid plasma. The heavy element at the bottom is called the erythrocytes. What sits above this are the white blood cells or leukocytes, platelets, and cell fragments, which are also known as thrombocytes. Leukocytes are larger than erythrocytes, but there are fewer in number. Most of their work involves the tissues, but they use blood as a transport medium. They also have many functions, but within the tissue spaces, they provide defense against organisms that can cause disease. Thrombocytes are small fragments of large cells called megakaryocytes. These cells develop from hemocytoblasts in the red bone marrow. Thrombocytes become sticky and clump together to form platelet plugs that close tears in blood vessels. They can also initiate the formation of blood clots. So the second layer is also known as the buffy coat due to it being a pale color. It also consists of less than 1% of a blood sample. Above this layer is the blood plasma, which is also pale in color but has a more yellow hue to it. The erythrocyte volume after being in the centrifuge is known as packed cell volume. In normal blood, about 45% of it is erythrocytes, The hemocrit of the sample can vary from 36 to 50%, and erythrocytes, leukocytes, and thrombocytes are the three formed elements of blood. There are many characteristics of blood, one being the color. Blood that has recently taken up oxygen appears a brighter red, and the blood that has recently released oxygen appears a more dusty red. This is because of the hemoglobin, which is a pigment that changes color depending on the degree of oxygen saturation. Next is viscosity. Blood tends to be sticky to touch, and its viscosity is about 5 times greater than water, which is just the measurement between a fluid's thickness or resistance to flow. And it is also influenced by plasma proteins and formed elements being present within the blood. This can impact blood pressure and flow. The temperature of blood is slightly hotter than our normal body temperature. Blood tends to be about 104 degrees Fahrenheit, whereas our normal body temperature is 98.6 degrees Fahrenheit. The pH of blood is roughly 7.4, but this can range very slightly. Blood tends to be more basic on a chemical scale and contains buffers that can help regulate blood if it drops a little higher or lower the pH scale it should be at. Blood is about 8% of the adult's body weight. Males usually hold about 5 to 6 liters of blood, whereas females hold about 4 to 5 liters of blood. So blood plasma. Plasma is composed of primarily water. This is about 92% water, and within this water is a mixture of substances, which are mostly proteins. Plasma proteins are about 7% of the volume of plasma, and there are 3 major groups of these plasma proteins called albumin, globulins, and fibrogen. The albumin is the most abundant and is manufactured by the liver. Their molecules serve as binding proteins, which are transports for fatty acids and steroid hormones. When lipids bind to albumin, it enables them to be able to transport in the watery plasma, even though they are hydrophobic. Albumin also contributes to osmotic pressure of blood, and its presence holds the water inside blood vessels and draws the water from tissues across the blood vessel walls and into the bloodstream, helping maintain blood volume and blood pressure. This makes up about 54% of the total plasma protein content. Next, we have globulins. This is a heterogeneous group with 3 main subgroups, alpha, beta, and gamma globulins. Alpha and beta transport iron, lipids, and fat-soluble vitamins to cells like albumin. They also can contribute to osmotic pressure. Gamma globulins are proteins involved in immunity, which are also known as our antibodies. They are produced by specialized leukocytes known as plasma cells. Globulins make up about 38% of our total plasma protein volume. So next we have fibrogen. This is produced by the liver and is essential for blood clotting. This accounts for about 7% of the total plasma protein volume. So other than those 3, we also have other plasma solutes, such as electrolytes, dissolved gases, organic nutrients, and metabolic waste. These non-protein solutes combined are about 1% of the total volume of plasma. So more about erythrocytes. As stated before, these are red blood cells and are the most common formed element. They make up about 25% of body's total cells, so they are quite small in size. Their functions include picking up inhaled oxygen from the lungs and transporting it to the rest of the body's tissues. It also can pick up carbon dioxide waste from the tissues to transport to the lungs to be exhaled. So erythrocytes have a unique shape and structure. They mature in red bone marrow, extruding its nucleus and other organelles. During the first day or 2 in circulation, an immature erythrocyte known as a reticulocyte will contain remnants of organelles. These should comprise of 1 to 2% of the erythrocyte count and provide an estimate of how many red blood cells are being produced. Erythrocytes are biconcave discs, meaning they are plump on the outsides and more thin in the center. Since most lack organelles, there is more interior space for hemoglobin molecules to transport gases. In the capillaries, the oxygen that is carried by erythrocytes diffuses in the plasma and then through capillary walls to reach cells. To travel through the capillary beds, erythrocytes may fold themselves to be able to fit through the narrow passages. So the life cycle of erythrocytes. Erythrocytes are produced at a rate of more than 2 million cells per second, but we need certain elements for this to occur, such as nutrients, glucose, lipids, amino acids, and trace elements. These trace elements are the following: heme iron, which is from animal foods and absorbed more efficiently than non-heme iron, which is from plant foods. When absorbed, iron becomes a part of the body's total iron pool. Bone marrow, liver, and the spleen store the iron in protein compounds called ferritin and hemosiderin. Copper is another trace mineral needed. It is a component of 2 plasma proteins, and without these proteins, hemoglobin could not be produced. Zinc is a trace mineral needed as kind of like a coenzyme that facilitates synthesis of the heme portion of hemoglobin. And lastly, we need vitamin B. These function as coenzymes as well to facilitate DNA synthesis. This is critical for synthesis of new cells, including erythrocytes. So erythrocytes live about 120 days in circulation, which when worn out, a myeloid phagocytic cell called a macrophage removes them. Degraded erythrocytes, hemoglobin is processed by globin, which is the protein of hemoglobin. This is broken down into amino acids, then sent to bone marrow to produce new erythrocytes. The iron within the heme portion of the hemoglobin can be stored in the liver or spleen and can also be recycled into new erythrocytes. The non-iron portion of the heme is degraded into waste called biliverdin and then into another waste called bilirubin. So next, we're going to talk about hemoglobin a little bit. Hemoglobin is a large molecule made up of proteins and iron. It consists of 4 folded chains of protein called globin. Each globin molecule is bound to a red pigment molecule called heme, which consists of an ion of iron. In the lungs, hemoglobin picks up oxygen, which will bind to the iron ions, forming oxyhemoglobin. The bright red hemoglobin travels to the body's tissues to release oxygen molecules, which gives the darker red color, which is also known as deoxyhemoglobin. Sorry, I didn't mean to click next yet. And then in the capillaries, carbon dioxide enters the bloodstream. 23 to 24% of this binds to amino acids in hemoglobin, which forms a molecule called carmaminohemoglobin. From the capillaries, hemoglobin carries carbon dioxide back to the lungs to be exchanged for oxygen. This binds to albumin and travels through the blood to get to the liver to then become bile. So next, we're going to talk about platelets. As stated before, platelets are also known as thrombocytes. A platelet is a fragment of the cytoplasm called a megakaryocyte that is surrounded by a plasma membrane. Platelets are relatively small in diameter with about 150 to 160 units of blood. After they enter circulation, about 1/3 migrate to the spleen to be stored to be released later when a blood vessel ruptures to limit blood loss. They are also critical for hemostasis, which is the stopping of blood flow damage to a vessel. They secrete a variety of growth factors for tissues to grow and repair as well. Hemostasis. Hemostasis is a process to stop bleeding from resulting in vessels. Platelets play a huge role in hemostasis. They typically help with smaller ruptures in 3 steps: the vascular spasm, the formation of a platelet plug, and coagulation, which is also known as blood clotting. First is a vascular spasm. This occurs when a vessel is damaged. The vascular system eventually occurs. This is when the smooth muscle in the walls of the vessel contract to constrict blood flow. Next is the formation of a platelet plug. This is the second step, where the platelets enter the damaged vessel. They clump together to become sticky and bind to the exposed collagen. They begin to release chemicals to start hemostasis, but this is only a temporary fix to the damaged vessel. Next, we have coagulation. Coagulation is the formation of a blood clot made up of fibrin, which is an insoluble filamentous protein derived from fibrinogen. Clotting factors that are involved in collagen are reactions to activate like more collagen factors. This process takes place in two pathways: the extrinsic pathway, which is triggered by trauma, and the intrinsic pathway, which begins in the bloodstream and is triggered by internal damage to the wall of the vessel. Both of these pathways merge into the common pathway, and these three pathways depend on clotting factors such as vitamin K. This clotting factor is secreted by the liver and platelets, which vitamin K is also needed to produce. Vitamin K is consumed in our diet and is also synthesized by bacteria that are in the large intestine. So a little bit about the intrinsic pathway. This begins when tissues become damaged, when in contact with blood plasma, the damaged cells that are also extrinsic to the bloodstream release thromboplastin, eventually forming an enzyme complex. The enzyme complex will activate the common pathway. Intrinsic pathway, this is kind of more complex, and the factors are now intrinsic to the bloodstream. This can begin with damaged tissues or when factor XII encounters foreign materials. So the common pathway, like stated before, both pathways lead to the common pathway where fibrin seals off the vessel. When factor X is activated by one of the two pathways, prothrombin is converted into an active enzyme called thrombin. Thrombin then converts fibrogen before the fibrin clot can become stabilized. So next, I'm going to talk about fibrolysis a little bit. This is when the proteins contract to pull on fibrin threads, tightening the clot together. This can cause fluid to be produced called a serum, which is made up of blood plasma. To continue normal blood flow, the clot must be removed. Fibrolysis is what degrades the clot slowly, and while this process takes up place, and plasminogen is converted into plasmin to break down the fibrin of the clot itself. So plasma anticoagulants. An anticoagulant is pretty much any substance that opposes coagulation, which can help limit coagulation in general. And this is my citations. I thank you for listening, and that is the end of my presentation. Thank you.